Today's presentation is Therapeutic Proteins, Drug-Drug Interactions, Updates and Challenges. Our speaker today is Dr. Khalid Ben-Kali. Khalid is currently a Director of Clinical Pharmacology at Certara. By training, he's a pharmacist and holds a Master's of Science and a PhD in pharmacology, as well as an executive MPA. Khalid has more than 10 years of experience in the pharmaceutical industry, where he previously worked at Galderma and Pierre Fabre, where he participated actively in the development of new chemical entities and monoclonal antibodies. Welcome to the presentation, Khalid. The floor is yours.
Thank you very much, Suzanne, for the nice introduction. So for this webinar, I wanted to share with you a topic which is under the spotlight since years. It is the drug-drug interactions for therapeutic proteins. A lot of things are happening and a lot of updates also are available from the regulatory agencies and also from the scientific community and the pharmaceutical companies. Before starting, I wanted first to start with some basic definitions about therapeutic proteins. So therapeutic proteins are protein-based drugs which may have enzymatic or regulatory activity, such as replacing a protein or augmenting an existing pathway or providing a novel function or activity. Therapeutic protein may have also special targeting activity, such as interfering with the molecular organism or delivering other compounds and protein. And also therapeutic proteins are used as immunizing entities, such as in the vaccines. In terms of molecular types, the monoclonal antibodies are the widely used and the more representative of this class of drugs and are accounting for a significant proportion of marketed and approved therapeutic proteins. But there are also other molecular types, such as the Fc fusion protein, anticoagulants, blood factors, etc. If you would like to know more about the clinical pharmacology of therapeutic protein, you can have access to my previous webinar that can be found on the Certara website. And this webinar presents the general information about the clinical pharmacology and the aspect to consider during the drug development of this class of compounds. Therapeutic proteins, and more specifically monoclonal antibodies, are now part of a standard therapeutic arsenal. They are widely used, and we can find a lot of combinations and concomitant use in the daily life of patients. So you may combine or have concomitant use of therapeutic proteins or therapeutic proteins with small molecules. So in this case, the drug-drug interaction is a real risk. This is not something which is exceptional or cannot be found in only in rare occasions, but could be encountered for every patient in every day. So as this risk is important, it is important to go through this risk during the development. What is also important that the DDI may occur between therapeutic protein and the small molecule. It could also occur between therapeutic protein compounds themselves. And the therapeutic protein could be both a victim, but could be also a perpetrator and responsible for a DDI. There are a lot of and several mechanisms of DDI. We'll go through some of them in this presentation, but you need to keep in mind that these are quite complex, and there is an additional layer of complexity that we don't have with the small molecules, is what we call the mediator role of the disease state, the immunogenicity of the target physiology. In terms of risk evaluation for the DDI, this is based on a good understanding of the clearance mechanism, the disease state, but also the pharmacology and the mechanism of action of the therapeutic protein, and also, of course, all the co-administered medications. In this webinar, I will focus on the most important aspects involved in the mechanism of the drug-drug interactions, which are the clearance mechanisms and also the disease state. So for the DDI involving impact on clearance mechanisms, therapeutic proteins are eliminated mainly through proteolytic degradation that results in smaller peptides and amino acids. And there are typically three clearance pathways in the elimination of the therapeutic proteins. The first one is a non-specific clearance, which is a target independent, and go through a non-specific endocytosis in cells, and also proteolysis in liver and the reticuloendothelial system. The second clearance pathway is a target-mediated specific clearance. So this is specific to antigen-specific interactions. So therapeutic protein antigen complexes are internalized and catabolized through lysosomal degradation. However, for this clearance pathway, the target can be saturated and can limit the capacity of elimination of the drug. If the target-mediated specific clearance is the most predominant clearance for a therapeutic protein, this may result in a specific profile called target-mediated drug disposition, or TMDD. And for drugs with TMDD, they are characterized by a non-linear PK profile. The third pathway of the clearance is related to the immunogenicity response, which may be developed in some patients developing anti-drug antibodies, ADA, and this ADA will form immune complex with the therapeutic proteins, and they are cleared via Fc gamma mediated endocytosis, and generally these complexes are eliminated at a different rate than the therapeutic protein alone. Overall, all these clearances may be present for the same compound at the same time, but their involvement is different, and this will define the overall clearance of a therapeutic protein. Overall, all these clearances may be impacted by the other drugs and may result in clinically meaningful drug-drug interactions. So now we can see examples of impact on each of these clearance pathways. So the first one about the non-specific clearance. Generally, the drug-drug interactions may be seen on an impact on a neonatal Fc receptor, or FcRn. So this receptor plays an important role during pregnancy for the transfer of passive humoral immunity from the mother to her fetus. And throughout life, this receptor protects IgG from degradation, which explain why this class of antibody has a long half-life. And this extended half-life is thanks to FcRn-mediated recycling, meaning that the IgGs are recycled when they are bound to the FcRn. So any drugs that can block, interfere with the FcRn may disrupt this recycling cycle and will increase the clearance of the drug and result in drug-drug interaction. So this may be seen for any therapeutic protein containing this Fc region of human IgG, which can bound to FcRn. This was, for example, observed when there is a concomitant treatment with the IVIG. So IVIG are commercial preparation of polyvalent human IgG. They are used at a very high dose in the treatment on some diseases. And if there is a concomitant treatment with other drugs, we may see some interactions. This was described, for example, for eculizumab, which is a monoclonal antibody targeting the complement C5 element. And you can see in this example that the combination of eculizumab with IVIG resulted in a significant decrease of the exposure to eculizumab in comparison to treatment without IVIG. And this is explained by the competition to the FcRn binding between the eculizumab and the IgGs that go through the IVIG administration. So there is no recycling of the monoclonal antibody, resulting in higher clearance, thereby resulting in lower exposure. Second DDI on the impact on the target-mediated specific clearance. This may be observed for drugs with the TMDD profile, meaning the clearance is mainly driven by the expression of the target. So if you modulate the expression or the concentration of the target, you may also modify the clearance of this drug. So this is the case for evolocumab. This monoclonal antibody was approved for lowering cholesterol, and it is targeting the PCSK9. So PCSK9 may be also modulated by the treatment with the statins. And in this case, if there is a co-treatment between evolocumab and statins, there is an increase of the PCSK9 levels, resulting in increase of the clearance of evolocumab by about 20%, and this resulted in a decrease of the exposure of evolocumab. The third impact, the impact on the ADA-mediated clearance. This may be observed when there is a co-treatment with immunosuppressant drugs. So immunosuppressant drugs may affect the immunogenicity profile, and if the drug is also eliminated by the ADA-mediated clearance, if there is a modification of the immunogenicity, we may also see a modification of the drug's clearance. There is a good example of that. It's the co-treatment of adalimumab with methotrexate in patients with axial spondyloarthritis. So in this example, the methotrexate reduced the proportion of patients with the positive ADA, reduce also the proportion of patients with high titers of ADA, so high titer will bring higher impact on the clearance. And so overall, this resulted in a higher exposure to adalimumab when the patients are receiving methotrexate, as you can see in this plot. Now we have reviewed the potential DDI when impacting the clearance of the drug. Now we'll move to the impact of the disease state, and more specifically, the pro-inflammatory cytokines-related drug-drug interactions. Pro-inflammatory cytokines such as IL-6, IL-1β, TNF-alpha, or interferon-alpha can downregulate the expression of cytochromes enzymes. And this will decrease the metabolism of the drugs that are CYP substrate, so this will increase their exposure level. However, the pro-inflammatory cytokines level may vary from a disease condition to another, as you can see in this example of the IL-6 levels or the TNF-alpha level in the different diseases. So we have different levels of these cytokines depending on the disease. And this will result in a different impact on the CYP substrate. If we take the example of the HIV patients with cachexia, so as we have higher levels of IL-6 or TNF-alpha, the impact on the CYP substrate was observed to be moderate, meaning that 2 to 5 fold impact on the AUC of the cytochrome substrate. However, in the case of the cancer, where the cytokines level are lower than what we can observe for other disease, so in this case, the impact on the CYP-sensitive substrate was considered as low or weak. This was lower than 2-fold impact on the AUC. So overall, any therapeutic protein that can modulate the pro-inflammatory cytokines levels will modify the regulation of the CYP expression, and this will modify the CYP activity and also the exposure of the CYP substrate. So this is what we call a disease-mediated drug-drug interaction. In this slide, you have an example describing how these drug-drug interactions occur. So here in this plot, you have the concentrations time profile of simvastatin, so a drug used to lower cholesterol. So these are the typical levels in healthy subjects, of course, except for their cholesterol level. So let's assume that those subjects develop an inflammatory disease like the rheumatoid arthritis. So in this case, the inflammation will result in the increase of the pro-inflammatory cytokines, resulting in a decrease in the CYP expression and activity, and this will result at the end in an increase of the CYP3A substrate, which is simvastatin. As you can see here, so the simvastatin level will increase significantly when there is this inflammatory disease. Now, if those patients are treated for their inflammatory conditions using drugs such as tocilizumab, an anti-IL-6, so this drug will lower the pro-inflammatory cytokines and then will normalize the exposure of the, will normalize the CYP activity, and this will result in simvastatin levels returning to levels close to what we observed first in the healthy subjects. So these were the most important drug-drug interactions mechanisms that are described in the literature, and the most important one to focus during drug development. Now, how to assess the DDI for these therapeutic proteins? When you compare to small molecules, the first approach is to start with in vitro assessment using a well-described and structured approach, using different models like the microsome, the hepatocyte, etc., to assess the risk before going to clinical investigations. Unfortunately, this is not always feasible and reliable for therapeutic protein. You saw that the DDI mechanisms are quite complex. They are also diverse, so a single approach is not appropriate. Some attempts were made to use hepatocyte models to assess the effects of cytokines on the CYP enzymes. Unfortunately, a lot of limitations were identified, including the variability of this model, and also this model was considered as a simple model in comparison to a very complex disease. So generally, you cannot take in account the complexity and the interactions between the different cytokines and the immune cell that could be found in inflammatory disease. So now it is acknowledged by the scientific community, but also by the regulatory authorities, that clinical evidence are preferred for informing the DDI risk assessment. For regulatory considerations, there are several guidance now available describing the approach and the importance to assess the drug-drug interactions for the therapeutic proteins. So the FDA issued the draft guidance in August 2020. This draft guidance provides a well-comprehensive approach how to evaluate the DDI and the risk, and describing all the approach how to conduct the clinical evaluation. On the other hand, the EMEA, the ICH also provide important high-level insight on the DDI assessment for therapeutic proteins. In the FDA draft guidance, you can find this very useful decision tree on how to assess the DDI, how to involve the labeling for therapeutic protein. So typically, the FDA guidance is classifying the DDI mechanism in two categories. The first one includes the pro-inflammatory cytokine mechanism, and the second one, in fact, is including all the other mechanisms, meaning that there is no one single mechanism in this one, but they are regrouping all the other situations in this category. So for the pro-inflammatory cytokine, if your drug is a cytokine, so in this case, there is no really question. You need to conduct a clinical DDI study to evaluate the risk for this therapeutic protein. However, in case of pro-inflammatory cytokines modulators, so the FDA is offering the possibility to include a labeling language informing about the potential interaction and its impact on the sensitive drugs, and more specifically for the drugs with a narrow therapeutic index. If not, you need either to provide robust scientific justifications to not include this labeling, or to conduct a clinical DDI study. For the other categories, so this will be really a case by case depending on the suspected mechanism. So they are suggesting or recommending to conduct a clinical DDI study to assess those risks without any precision for this case, because this will be quite diverse and complex situations. And in any case, the results of the clinical DDI studies need to be described in the labels of the drugs later. For the clinical studies considerations, the first question about which population to include in those studies. Generally, for small molecules DDI studies, we enroll healthy volunteers in the clinical studies, which is convenient and quite easy to recruit for the evaluation of the DDI. However, as you saw, most of the mechanisms described here may involve some specific mechanism related to the disease itself. It is the case, for example, for the cytokines modulation. So cytokines levels are very specific to a disease and some severity to the disease. So you need to enroll the right patients to evaluate DDI. And it is also the case for the TMDD interactions, where you are evaluating the DDI depending on the level of your antigen. So this also may be found only on the patients and not in healthy volunteers. In terms of the study design, there is no strong recommendations, but this will depend on the drug characteristics and also the suspected mechanism. So parallel design may be appropriate when you are evaluating the therapeutic protein as a victim. This is the case, for example, for the monoclonal antibodies, because they have quite a long half-life, so you cannot have sufficient washout between the two administration of the drug, so only parallel design is feasible. However, intra-individual design could be an option when you are evaluating the therapeutic protein as a perpetrator, when you are evaluating the impact on CYP substrate. So these are small molecules with a short half-life, so you can easily have a washout between the two administrations. In terms of study duration, this will also depend on the therapeutic protein half-life and also the time course of the involved physiological process. So there is a time necessary to suppress the inflammatory status, or for example, to modulate the antigen. And generally, this results with quite a long the duration of the study could be longer than we can see for small molecules. For the timing of the DDI studies, so when you have to conduct those clinical studies, so generally this will depend on the criticality and the impact of the DDI, but generally it is acceptable to conduct such studies in parallel to the phase 3 program, because these data are needed to be filed with a dossier for the BLA dossier to inform later the labeling. In this slide, you have a typical design of a DDI study involving pro-inflammatory cytokines DDI. So this study was conducted for risankizumab, an anti-IL-23 approved for the treatment of plaque psoriasis. So in this study, they enrolled the relevant disease population, the moderate to severe chronic plaque psoriasis patients. In terms of the design, they used a single oral doses of sensitive prop substrate using a cocktail approach. And these cocktail drugs were administered before starting the treatment of the therapeutic protein and after the treatment with the therapeutic protein. So there is a sufficient time for the washout between the two administrations. For the study duration, this was driven by the treatment duration with the risankizumab, which was 12 weeks, and this was necessary to achieve the therapeutic objective, meaning the treatment of psoriasis, but also the cytokines modulation. And in this example, you can see that no clinically meaningful drug-drug interaction were observed when using this treatment. In this table, you have the summary of the recently approved cytokines modulator and how the labeling was prepared for these drugs. So you can see that for these examples, the labeling were consistent with the FDA recommendation, meaning that for some drugs, they didn't conduct any clinical DDA studies, but they included a specific labeling language to inform about the potential DDI and its impact on the other drugs. On the other hand, there are other drugs. They conducted the specific clinical studies using the CYP cocktail approach, and then they described the study results in the labels. The modeling and simulation could be a very interesting and useful tool to assess the impact of the DDI of therapeutic proteins, but also could be used to predict what could be the risk for such DDI. So you can use the standard approach of population PK modeling and simulation. So this can be used to evaluate the victim potential of the therapeutic protein. So for example, during the covariate search to identify the impact of other concomitant medication. On the other hand, you can also use this approach to evaluate the perpetrator potential of the therapeutic protein. This is, I would say, more difficult and complex, but feasible. You need just to have a prospective planning and proper study design to collect the right data from the other SIP substrates to evaluate this. This is not always done, but you can plan it in advance and do it during your population PK modeling. On the other hand, the mechanistic approach could be also very interesting. So there is one recent interesting example for the DDI risk assessment for a antibody drug conjugate, so ADC, which is a payload grafted to a monoclonal antibody for the treatment of cancers. And in this example, they used the PBPK model to inform the DDI risk of polatuzumab vedotin without conducting the dedicated clinical trial. So the results of this modeling was included in the label to inform about the risk of the DDI. Hopefully, there is more to come in the near future to simulate this cytokine-related DDI mechanism. And this could be very interesting in the future. So without conducting specific clinical trials in patients to evaluate the DDI. So maybe you can use the mechanistic modeling for that. So my colleagues from Simcip are working hardly to develop such tools, which probably will be included in the next version of Simcip, the version 23. So in this tool, the objective is to simulate simultaneously the impact of the therapeutic protein on the small compounds through the modulation of CYP expression. Overall, the approach is to have this PBPK model for therapeutic protein and small compounds and take in account the cytokines modification and its impact on the PK of the small molecule. And thanks to the PBPK model, you can predict what could be the impact on the pharmacokinetic profile of the small molecule. So this approach was successfully used, as you can see that the predictions using this model, in this example, it was for IL-6 modulation, and all the predictions were consistent with the observed and the published data about these simulations. Now to conclude this webinar, you saw that the drug-drug interactions involving therapeutics protein are likely and should be considered during the drug development. So this is something you need to do for every therapeutic protein development. The DDI for therapeutic proteins involve diverse and complex mechanisms of actions. And for accurate risk assessment, you need to understand well what are the clearance mechanism, the effect and the impact of the disease state, and also to have a good understanding of the pharmacology and the mechanism of action, and to evaluate all the co-administered medications if there are systematic medications or maybe just a concomitant use. Now there are regulatory guidelines providing important information and a comprehensive approach how to assess the DDI risk, how to conduct the clinical investigations, and also how to inform the labeling for these drugs. And finally, you saw that the mechanistic modeling is really a promising tool in the future to assess the complex therapeutic protein DDI risks. So this concludes this webinar, and I'm happy to answer any question. Thank you very much for your attention.
Great, thank you so much, Khaled. And we would love for our audience to submit their questions to him. So it looks like we've got a couple questions already. Someone would like to know if you're aware if any of the checkpoint inhibitors or other immune modulators were evaluated for drug-drug interactions with regard to CYPs, or are these regarded as locally acting with respect to cytokine levels?
Yeah, very interesting question. In fact, there are some recent publications from the Simcip team about these drugs, specifically for the cytokine storm, where there is a release of the cytokines, which could also be considered as a modulation of the cytokines. So this will act in a different way than what was described in this presentation, meaning that we will not decrease the cytokine by the treatment, but in contrary, it will be in the other side. And in the same way, for example, there are some investigations for the COVID, which may also result in a significant increase of the cytokines.
All right, thank you. Hopefully this question's clear. If not, we can ask the person who asked it for clarification. What is the difference between a pro-inflammatory cytokine modulated therapeutic protein and a pro-inflammatory cytokine therapeutic protein?
So the modulator are generally monoclonal antibodies that are targeting a specific cytokine or specific receptor. So this will interfere with the cytokines in the body, where the cytokine therapeutic protein, meaning that you are using cytokines. This is something, for example, used in some disease in oncology, where the cytokines are considered as a drug, ever to stimulate some pathway or to interfere with some pathway.
Great, thank you. Got a lot of questions coming in, which is fantastic. Someone says, great presentation, I agree. Can you touch upon doing CYP induction assays for therapeutic proteins?
So if I understand the question is about the in vitro assessment of CYP induction. In this case, as I presented for the in vitro models, the hepatocyte, some attempts were made. Unfortunately, these models cannot be really reliable. And in terms of clinical aspect of the induction, we saw that in the modulation could be in both ways, meaning that the modulator can decrease the CYP expression, but there are other drugs like the check inhibitors resulting in a cytokine storm, which can also induce the cytokine on the other side. I'm not sure that I'm answering correctly the question, but please feel free to reach out to me later.
I think, yeah, the person responded that you did answer it, so thank you. There are a number of combination therapies in oncology involving co-administration of IgG monoclonal antibodies, for example, CPIs with TIGIT or LAG3. Has the agency, and we'll just assume that means FDA, has the agency asked for PK studies of the individual proteins in these presentations?
I'm not sure that I am aware about all the situations for oncology DDI, but what is important is to present to the agencies first a risk assessment of the DDI. So I think this is something very important and needed. So you need to consider for all these combinations if there is any chance or if there is a risk that you will see a drug-drug interactions. The second step will be to evaluate if this risk is really important for the patients. Is it impacting the safety? Is it impacting the efficacy? And then you need to figure out if a clinical study is needed or not. So I would say really this would depend on the situation. It will be case by case. But at least for oncology, for those drugs, I'm not sure there are studies for every situation. As it is now the case, for example, for small molecules in oncology, where the DDR start to be something like a standard approach to evaluate the DDR for small molecules. For the therapeutic proteins, I'm not sure that we have studies for every situation, but at least the evaluation of the risk is something very important.
So when you talk about therapeutic proteins, you talk about monoclonal antibodies, you talk about antibody drug conjugates. Do you consider mRNA-based vaccines to be therapeutic proteins? They're not proteins. They're nucleotides.
Yes, this is a good question. I would say yes. Just the mRNA are really in between the small molecules and the therapeutic proteins. Depend on the I would say the manufacturing process. But the objective of the mRNA vaccine is at the end to synthesize the therapeutic protein. So what is important is the effect of the therapeutic protein. So I would say yes, they are considered therapeutic proteins.
Okay, great. On slide 20, you had a list of monoclonal antibodies that were studied where, and you say, someone asked us about the monoclonal antibodies where drug-drug interaction studies weren't conducted and they got labeling as there being a potential drug-drug interaction risk. Are those monoclonal antibodies required, or are those drug programs for those monoclonal antibodies required to perform post-approval DDI clinical studies?
The answer is no. As you saw in the decision tree, the agency is offering the possibility that the sponsor, the applicant, can add the appropriate labeling language to inform the prescriber about this risk and the potential DDI. In my opinion, this is feasible because the extent of the DDI is relatively limited, could be low to moderate. So we know it, we know that there is potentially interactions. We are informing the prescriber about this risk. And when you see the exact language, there is a specific focus on some drugs with the narrow therapeutic index, but there is no specific recommendation to have this as a post-marketing commitment as far as I know.
Great. So someone has a question about antibody drug conjugates, and they want to know how often are drug-drug interaction evaluation needed for the ADC payload when the payload acts as a perpetrator? And they say, are some known to be more likely to cause DDIs than others? Example, DNA modulators versus tubulin disruptors, or are these payloads more likely to be DDI victims?
So for the ADC, here we are in a hybrid situation between the therapeutic protein and the small compounds. So for the example I was talking about, I think the risk is more related to the payload component, which is this small molecule. So for this one, you need to conduct all the in vitro studies to evaluate the impact on the different CIPs, transporters, etc. And then you need to follow the standard guidelines for small molecules, meaning that you evaluate the risk in regards to the circulating concentrations, having in mind that for these drugs, their therapeutic index is quite narrow. They're very toxic drugs, so it is important to evaluate the risk. But I think the regulatory agencies are aware of that. This is for oncology, so this is quite a specific area where DDI studies are not always feasible. Conducting those studies in healthy volunteers is probably not an option. So for this reason, I was saying that a PBPK modeling is a very interesting alternative to conduct a DDI study. But what is sure that the evaluation of the risk of the DDI is something mandatory for all the ADCs. Clinical studies are not, I would say, probably systematic for every situation, but you need to evaluate the risk. And now, as we have access to PBPK approach, I think we should use these approaches to inform the risk.
Fantastic. Someone says, thanks so much for the talk. You showed that drug-drug interactions mediated via therapeutic protein clearance mechanisms, but they all seem to have between 20 to 50% change, so the change is pretty limited. Would you say that victim DDIs, are they generally of this limited extent?
I don't have the exact answer. It's true that I tried to find another example with more significant, but I was not able to find it. This one for evoluculumab was an interesting one because when you look to their data, it was very nice. They evaluated the impact on the exposure to evolumab, but also they have data on the expression of the target. So they have the PCSK9 levels with statin, without statin, etc. So you can really make the link between the PCSK9 levels and the impact on the clearance. So for this one, this DDI was not considered as clinically meaningful, and this is clearly, I would say, available in the dossier of this drug, and it's not appearing in the label as a significant DDI. But I don't know if there is other examples. But for I would say for drugs that are under development, this is something very important to evaluate if there is an impact or not on the drugs which have a TMDD profile.
Okay, looks like we've got one last question, and this one may be for our Simcip colleagues or may not be. Someone asks about Certara's mechanistic DDI model. I'm assuming they're talking about Simcip. Where does the cytokine inhibit small molecule parameter come from? Does that come from in vitro or in vivo data?
For this one, probably I may to defer the question to my colleagues from Simcip because they are working on this, and these slides were a courtesy from them. But in fact, I think they are using both in vitro data, but they have access to some clinical data. But there is a nice summary of published recently by the Simcip team, publishing all the in vitro data that were used, and for sure we can share with you this article.
Fantastic. Well, thank you so much, Khalid, for that really interesting and educational program. Just want to tell folks a few announcements before we wrap up. Our next webinar in our scientific webinar series will be on January 31st, where my colleague, Dr. Mark Lovern, will present what's new in physiologically based pharmacokinetics in the Simcip simulator. So if folks had a lot of questions about Simcip, you should come to that webinar and learn what exciting developments have been made. You can register for this webinar on the Certara website. And with that, I'd like to thank our audience for their attention and their fantastic questions. On behalf of Certara, have a great day, and this concludes the webinar.